Hi everyone, and welcome back to All Things GC. Today I'm doing a review of mosaicism. Mosaicism occurs when an individual has two or more genetically distinct cell populations that originated from the same fertilized egg. That's an important distinction. Because all of these cells came from the same zygote, they all started with the exact same DNA. At some point after fertilization, however, a genetic change occurred in one cell. As that cell continued dividing, every daughter cell inherited that genetic change, while the rest of the body continued developing normally. The result is an individual with a mixture or mosaic of genetically different cells. Think of it like a branching family tree. Imagine a mutation occurs very early in development, when there are only 8 cells. Everything descending from that one abnormal cell carries the variation. If the variation instead occurs much later, perhaps after millions of cells have already formed, only a very small region of the body may contain those abnormal cells. So one of the biggest factors determining the extent of mosaicism is when during development the variation occurs. Generally speaking, earlier variations affect more tissues. Later variations affect fewer tissues. That simple concept explains many of the clinical differences we see. Before we go further, let's distinguish mosaicism from another concept students often confuse it with: chimerism. Both involve multiple genetically distinct cell populations, but they arise in completely different ways. In mosaicism, every cell originated from one fertilized egg, and the genetic differences developed after conception. In chimerism, the genetically different cell populations come from two different zygotes. Chimerism examples include twin-to-twin cell exchange, fusion of two embryos early in development, organ transplantation, and bone marrow transplantation. So remember, one zygote equals mosaicism, two zygotes equals chimerism. Now, how does mosaicism occur? There are several mechanisms. The first is a postzygotic mutation. After fertilization, DNA replication isn't perfect. A mutation can occur during any cell division. This could be a single nucleotide variant, a deletion, a duplication, a chromosomal rearrangement, a repeat expansion, or essentially any other genetic alteration. Once that mutation occurs, every cell inherits it. Another common mechanism is mitotic nondisjunction. Most students remember nondisjunction from meiosis, where it causes trisomy 21 or trisomy 18. But nondisjunction can also occur during ordinary mitotic cell division after fertilization. When sister chromatids fail to separate properly, one daughter cell gains an extra chromosome while the other loses one. This produces chromosomal mosaicism. Another mechanism you'll hear about is anaphase lag. Instead of chromosomes failing to separate, one chromosome simply fails to make it into the daughter nucleus and is lost. This often results in one cell becoming monosomic while the other remains normal. Anaphase lag also explains some cases of trisomy rescue, which we'll discuss shortly. Now, timing is one of the highest yield concepts we'll cover. Imagine a variation occurring at the two-cell stage. Half of the body's cell may eventually carry that alteration. Now imagine the exact same variation occurring several weeks later. Perhaps only one part of one organ carries it. The earlier the variation occurs, the higher the proportion of affected cells, the more tissues are involved, and the greater the likelihood of clinical symptoms. Later variations generally produce lower levels of mosaicism, more localized disease, and sometimes no symptoms at all. This is why two individuals with the same variation can have dramatically different clinical presentations. Next, let's discuss two major categories. The first is somatic mosaicism. Somatic mosaicism affects body cells. These individuals may have variable disease severity depending on which tissues are affected, how many cells carry the variation, and whether those tissues are clinically important. Importantly, somatic mosaicism may not be inherited if reproductive cells are unaffected. The second is germline mosaicism. In germline mosaicism, the mutation is present within some egg or sperm cells. The parent is often completely healthy because their body tissues either don't carry the mutation or carry very little of it. However, they can still have multiple children with the same dominant disorder. This explains an important counseling scenario. Parents have one child with what appears to be a de novo pathogenic variant. A second child is born with the exact same pathogenic variant. Rather than two independent de novo events, one explanation is parental germline mosaicism. This is why we usually avoid saying recurrence risk is absolutely zero after a de novo diagnosis. There is often a small residual recurrence risk because of possible germline mosaicism. Now let's focus on chromosome abnormalities. One of the most common examples is mosaic Down syndrome. Instead of every cell containing 3 copies of chromosome 21, only a proportion of cells are trisomic. Clinical severity can be highly variable. Some individuals resemble classic Down syndrome. Others have relatively mild findings. Importantly, the percentage of trisomic cells in blood does not necessarily predict severity, because blood may not reflect the percentage present in the brain, heart, or other organs. This is an important counseling point. Blood is simply one tissue sample. It doesn't always represent the entire body. Another high-yield mechanism is trisomy rescue. Imagine a fetus begins as full trisomy. Early during development, one of the 3 chromosomes is lost through anaphase lag. Some cells return to the normal chromosome number. Other cells remain trisomic. The result is chromosomal mosaicism. Trisomy rescue is also clinically important because it can provide uniparental disomy, depending on which chromosome is lost. Confined placental mosaicism is our next topic, and this falls into prenatal genetics. This is one of the most important concepts. It occurs when the chromosome abnormality is present in the placenta but absent from the fetus. Because cell-free DNA screening analyzes placental DNA, not fetal DNA, confined placental mosaicism is one reason we can obtain a positive screening result, even when the fetus has normal chromosomes. This is one of the classic explanations for a false positive cfDNA screen. Historically, 3 major patterns of confined placental mosaicism have been described. Type 1, involving the cytotrophoblast, type 2, involving the extraembryonic mesenchyme, and type 3, involving both the cytotrophoblast and mesenchyme. Different types carry different implications for fetal involvement and pregnancy outcomes, but the key concept is simply understanding that the placenta and fetus do not always share identical chromosome complements. Even if the fetus is chromosomally normal, confined placental mosaicism has been associated with pregnancy complications, such as fetal growth restriction in some cases, which is one reason these pregnancies may receive additional monitoring. Now, how do laboratories identify mosaicism? It depends on both the technology used and the proportion of abnormal cells. One way is through karyotype. A standard chromosome analysis evaluates multiple metaphase cells. If only a subset of cells contain the abnormality, mosaicism may be identified directly. Increasing the number of cells analyzed improves the chance of detecting low-level mosaicism. Another is a chromosomal microarray. Microarrays detect copy number differences across DNA extracted from many cells. Moderate or high-level mosaicism is often detectable. However, very low-level mosaicism may fall below the platform's detection threshold. Fish can also be used. Fish can be particularly useful because hundreds of cells can be scored relatively quickly. This makes it valuable for confirming suspected mosaicism or evaluating specific chromosome abnormalities. Lastly, there's next-generation sequencing. NGS has greatly expanded our ability to detect mosaic variants. Instead of evaluating chromosomes, sequencing measures the proportion of DNA reads containing a variant. If approximately half the reads contain a heterozygous pathogenic variant, that suggests the variant is present in nearly all cells tested. If only 10% or 15% of reads contain the variant, mosaicism becomes a consideration. Coverage depth, tissue selection, and bioinformatic filtering all influence how reliably low-level mosaicism can be detected. One of the biggest clinical challenges is that mosaicism may not be present in every tissue. A blood sample may be completely normal while the mutation is present in skin, buccal cells, fibroblasts, brain, tumor tissue, or gonadal tissue. This is why negative blood testing does not always exclude mosaicism. Sometimes testing in other tissue is necessary if clinical suspicion remains high. So why does all of this matter? It matters because mosaicism influences nearly every aspect of genetics. It can affect disease severity, age of onset, diagnostic yield, genetic counseling, recurrence risk, prenatal interpretation, and cancer genetics. It also reminds us that a single blood sample represents only one tissue, and sometimes only one piece of the overall genetic picture. And that's your review of mosaicism. Hit that like and subscribe button for more genetics content, and I'll see you next time.